Thank you very much. Thank you very much, everybody. Can you hear me? The Pacific Northwest is particularly susceptible to ocean acidification, in large part because we live at the tail end of the Great Ocean Conveyor. Depicted in this slide here, the red band illustrates sort of the average flow of surface water towards the North Atlantic, where it sinks into deep water, and this deep water flows all the way back to the North Pacific. And as this water moves through the deep ocean's interior, bacterial respiration produces carbon dioxide. And that carbon dioxide, when it dissolves in water, produces carbonic acid. That carbonic acid loses hydrogen ions and is converted to bicarbonate and carbonate. And these hydrogen ions are what gives the seawater its acidity. So that by the time this deep ocean water reaches the North Pacific, it's already quite acidic. But then on top of that, seasonal upwelling off our coast helps to bring some of that deep water closer to shore, to the Strait of Juan de Fuca, where an analogous circulation pattern, estuarine circulation, which on average is out the top and in the bottom, can pull this acidic water up the Strait of Juan de Fuca, over the sill, in this case, which would be Admiralty Inlet, into the Sound. And as this deep water moves along the bottom of Puget Sound, elevated respiration rates produce carbon dioxide, which, guess what, makes the water even more acidic. These patterns were first quantified in a paper published a few years ago in 2010 by Richard Feely at NOAA. And these diagrams on the right side of the screen illustrate this. These diagrams are similar in geometry to this figure here, where we've got the bottom here. This is the Strait of Juan de Fuca. This is Admiralty Inlet, the sills, and this would be Hood Canal. And the colors indicate salinity, oxygen, pH, and aragonite saturation. Now, aragonite is the form of calcium carbonate that makes up clam shells and the shells of other calcifying organisms. And when this number is below 1, it means that dissolution of those shells is favorable. The important thing to learn from these diagrams is that as you move along the bottom of, say, Hood Canal, as we move, for example, to the south, oxygen concentrations drop, pH drops, which means the water gets more acidic. And the aragonite saturation state, which starts at about 1, drops to pretty low levels, about 0.5, which means Hood Canal has really corrosive seawater, enough to dissolve bivalve shells. However, a similar study, to my understanding, had never been conducted in Bellingham Bay until last spring, when senior undergraduate students at Western and my capstone course in Huxley College were challenged by me to do it, to measure these parameters in Bellingham Bay, find out what's going on, and assess what is causing acidification, if we indeed see it. Turns out they got some really interesting results. They found corrosive water, they assessed what's causing it, and they projected changes for the future. But before I show you those student data, I'd like to define a few terms so that we're all speaking the same language. I already mentioned that when carbon dioxide dissolves in seawater, it produces carbonic acid, which produces bicarbonate and these hydrogen ions. And that's called the carbonate system. The CO2 that dissolves in seawater then doesn't stay as a dissolved gas. The total carbon in the system, called dissolved inorganic carbon, is the sum of these components. The acidity of the system we can measure using sort of pH, which most of you remember from chemistry, drops as acidity goes up, right? And then alkalinity, which is a trickier concept, is sort of the buffering capacity of seawater. It's essentially a measure of all these anions that can suck up the hydrogen ions that cause the acidity, minus the hydrogen ions that are already there. But in English, it's kind of the buffering capacity of the seawater. And then finally, this last term, this aragonite saturation, is a way to describe how a certain pH level would affect shell-bearing organisms. That's essentially the ratio of the product of calcium and carbonate concentrations in seawater divided by what we would expect it to be at saturation. Again, in English, if this number is bigger than 1, it's good. If it's smaller than 1, then dissolution would be favored. Finally, all these parameters that I've rapidly defined actually are related to each other quantitatively through equilibrium chemistry. And in English, what that means is we can measure any two of these parameters and use a chemical equilibrium model to calculate the others. So in our case, at Western, we can measure dissolved inorganic carbon and pH with really high precision, and then use a chemical equilibrium model that's available to anybody, it's free, to calculate the other parameters. And that's what we did. I sent the students out on the boat. I went with them to make sure nobody fell over. And we sampled several stations in the bay. At each station we measured, we took water samples using a device called a CTD, which measures temperature and salinity and depth, along with water sampling bottles. And we collected samples at 6 different depths and measured the dissolved inorganic carbon, the pH. I had another group of students measuring the rate of respiration in Bellingham Bay bottom waters. Another group measured the sediment respiration. I had a group measuring these parameters in the Nooksack River. We got data from the wastewater treatment plant to try to understand not only what's happening in Bellingham Bay, but if we saw any acidic water, what's causing it. Whew. So with that in mind, let's look at some of these results. This is a plot of pH on the horizontal axis versus depth from these stations that we sampled. And note that pH is high in surface waters. Photosynthetic organisms remove carbon dioxide from the water, raising the pH. But at the bottom, where there's no light and no photosynthesis, respiration dominates. And that respiration, producing carbon dioxide, helps to lower the pH at depth. Now, our lowest pH was found right here at station C, which is about where this zone of hypoxic water, low oxygen water, tends to form seasonally. And our lowest pH was about 7.5. Now, to freshwater scientists, you'd think, that doesn't sound low at all. But in the ocean, it actually is. It's actually as low as you get in the acidic waters of Hood Canal. And to understand what that means in terms of biology, we can plot that aragonite saturation state right here. This is that, remember, that ratio that if it's bigger than 1, precipitation is favored. If it's less than 1, calcium carbonate shell dissolution is favored. And again, at the surface, it's really high. In other words, calcium and carbonate are supersaturated in surface waters in Bellingham Bay, in part because of the high pH. But at our site in station C, we get really, well, actually it's low all over, but right at this station, we get levels of about 0.5, which means the water is really corrosive to bivalve shells. It's every bit as corrosive as that Hood Canal water, where in the larger scale survey, we found really acidic waters. So this is the basic observation. The fact that this low pH, low aragonite saturation area occurs in the same spot where we get seasonal hypoxia suggests that maybe respiration plays a role like it does in the open ocean. So my students measured rates of respiration, and these plots show the respiration rate versus depth for these different stations in terms of oxygen consumption. And these rates are relatively high compared to average rates in Puget Sound. At this time of year, Bellingham Bay is really biologically active with fairly high rates of respiration, which may play an important role in the creation of this acidic water. But this is not the only place where respiration occurs, because it also occurs in the sediments. So we had another group of students collecting sediment cores. We incubated these sediment cores, and these incubation cores have stirring bars and sampling ports so that we can incubate them at near in situ temperatures and sample over time. And so we sampled the dissolved inorganic carbon and the pH. And what we found, this is a plot of the change in this total inorganic carbon, the DIC, in the sediment cores over time, and each color represents a different core. And the slope of this line gives us the sediment respiration rate. Interestingly, the respiration rates in the sediment are more or less equal to the total respiration in the overlying water. But there's one big difference. Respiration in the water column produces CO2 and it lowers pH, but it doesn't affect alkalinity. The reason is that as CO2 dissolves, it produces these components that increase alkalinity, like bicarbonate and carbonate, but it also produces hydrogen ions that equally reduce alkalinity. So there's no net effect on alkalinity. But in the sediments, in addition to this production of dissolved inorganic carbon that would lower the pH, sediments are also a source of alkalinity, an important source for the bay, it turns out, which tends to moderate the effects of the sediment on the overlying water chemistry. So the sediments and the water column act somewhat differently. But there are other sources of inorganic carbon to the bay. To link the processes that we studied in Bellingham Bay to the larger watershed, I sent another group of students to measure these same parameters at several points along the lower Nooksack River, Deming, Linden, and near the mouth of the Nooksack River where it enters Bellingham Bay. And what they observed is that as you go from towards the mouth of the bay, the dissolved inorganic carbon tends to increase. This little drop at the end is because my student took a sample at this really nasty little tributary. So we can kind of ignore this last point. But the general trend is increase in dissolved inorganic carbon, increasing alkalinity, but the alkalinity doesn't increase as much as the dissolved inorganic carbon, and so the pH actually drops as you get closer to the mouth of the Nooksack River. So the Nooksack River is supplying low pH water to the bay. But the other really important thing that the Nooksack River does is that it creates this density gradient in Bellingham Bay that drives this same two-layer circulation that we see in the rest of the sound, sort of southward in the surface water and northward in the deep water. So these are some of the patterns we observed. But what's causing them? What causes this corrosive water in the middle of the bay? Well, to address that, another group of students in my class constructed a simple model of the system. And here we break the bay into surface waters and deep waters. And the arrows in these boxes indicate the sources and sinks of dissolved organic carbon and alkalinity, and then you can calculate how that would affect the pH. And we have arrows coming in and out of the bottom box with respiration, sediment fluxes, inflow from deep water, mixing with surface waters, and so on. One problem is that there's a lot of stuff going on the surface, gas exchange and photosynthesis, that we actually didn't measure. It's very complicated. But fortunately, Bellingham Bay has a buoy pretty much in the middle of the bay that continuously measures pH in the surface water. So I had my students model the effects of all these sources of dissolved inorganic carbon and alkalinity north of the bay. So the surface water on average flows past the buoy, which measures the pH, so we don't have to calculate it, we just use the buoy measurement. And the students could then calculate how these processes affect pH and aragonite saturation, that is the effects on calcifying organisms in bottom water. So what did they find? Well, first of all, our data were sort of a snapshot at one point in time. But at this time period when we did the study, the bay was undergoing huge changes. This is the sort of domain of our model. The Nooksack River was varying, the surface pH was varying, but what does the model suggest it was happening to pH and aragonite saturation and the other components of the carbonate system? Well, the model suggests that at this time when we took a snapshot was a time when all these parameters were changing, that dissolved inorganic carbon was increasing, alkalinity was increasing, pH and aragonite saturation were dropping. In other words, we probably were measuring at a time when seasonal variation in these parameters were causing the bay to get more acidic. But what's causing this pattern? Well, my students varied parameters in this model. And one thing that they did is they removed water column respiration. This plot shows pH over the time period that we measured and this aragonite saturation over the same time period, measures of acidity. And when you remove water column respiration, you get very little change in these parameters. In other words, it looks like water column respiration is driving this low acidity conditions in the bay. Where am I? 2 minutes? Excellent. Finally, in 1 minute, what about the future? What about the big question of CO2 in the atmosphere? Well, to understand that, you have to calculate another parameter called the Ravel factor, which is a measure of how much CO2, for a given change in atmospheric CO2, how does that affect the dissolved inorganic carbon in the water column? So I had a group of students calculate that, and they added that to the model. And basically what they did is they said, well, let's imagine the scenario pre-industrial today, and with this future scenario that Bob Mitchell talked about, the business as usual scenario, let's calculate it 50 years into the future. And they essentially started the model with different levels of carbon in the system. The basic story is, if this is today, pH during this time period today, if all the other parameters are the same except for the CO2 in the atmosphere, this is what they expected pH was like pre-industrial. This is what they expected aragonite saturation was like in pre-industrial. In other words, acidic conditions not so bad. In the future, the models suggest a strong decline in pH due to increases in atmospheric CO2. The point is this. My students did find highly corrosive water in Bellingham Bay. It looks like the major reason for that is respiration in the water column combined with the sort of slow circulation. I didn't talk about it, but there's a circulation story as well. That is, as circulation sort of slows down, there's more time for CO2 to be produced and for pH to drop. And so slow circulation leads to more acidic conditions. Finally, we've heard all about reduced summertime flow of the Nooksack River predicted by global or regional climate models. That plus increased CO2 in the atmosphere together suggests that Bellingham Bay is likely to get more acidic. So we're at the tail end of the Great Ocean Conveyor, which starts us off at a disadvantage, and it looks like, sadly, it's going to get worse. The good news is that my students learned a lot. And we forged some great relationships, some great collaborations. Brooke Love, one of my colleagues, helped us with the dissolved organic carbon measurements and sort of thinking about this. Other technicians like Kelly Bright helped us with the pH measurements. Folks at the Shannon Point Marine Center, where we did a lot of this work, assisted us as well. So all of that was a great experience, despite the sad observation. But thank you for your attention.
Am I missing something from your raw data? What I'm seeing is pH of 7.4 or
yes.
higher.
Yes.
The last time I did chemical analysis, pH of 7 was neutral.
Yes.
And anything lower than that was acidic.
Yes.
So how do you contend that
yes.
your pH at 7.4 is
yes.
corrosive?
Because the ocean is not made of pure water. pH of 7 is neutral for pure water, but the ocean has a different alkalinity, has a whole suite of different solutes in it, which means it's a different beast. Now, that's why I meant to mention pH 7.5. Did I mention that wasn't a big deal? I think I intended to make a joke out of that, and I forgot to. But let me explain the reason for actually calculating this, both the pH and this aragonite saturation, is that the aragonite saturation state allows you to understand whether or not this pH is corrosive to organisms that have shells. And the answer is, in seawater, 7.5, given the alkalinity and other parameters in the system, is corrosive, and it will dissolve calcium carbonate shells. Now, turns out that adult bivalves can handle this, but this level of acidity can kill bivalve larvae, oyster larvae, for example. When it gets this low, they're in big trouble. And so that's the answer. It's not pure water, it's seawater. In fact, lake water and river water typically has pHs that are on the order of 5 to 6, and that's because of the CO2 dissolved in it, which produces carbonic acid. And so sort of neutral pH sort of depends upon the solution. That's an excellent point. Thank you for bringing that up. Other questions? Oh, yes, Brooke. Oh, no. The ocean acidification expert, Brooke Love.
No, I'm not gonna ask you any like terrifying questions. No, you mentioned that your students had measured like inputs from the wastewater treatment plant.
Yes.
And I was curious whether those were just like a tiny drop in the bucket compared to
Yes.
these other things. Yeah.
Yes. Thank you for asking that. We just collected data. They measure alkalinity and pH, and the flows are not significant enough, and it goes out the surface, so it didn't play a big role in the model, but thank you for asking that. The wastewater treatment plant, we've studied a lot, nutrient budgets and so forth, and it typically, because of the volumetric flow rate is low relative to like the Nooksack River and deep water coming in, it tends to play a small role in the overall function of the bay, at least so far when we've studied it in my classes. Yes?
Yeah, just got to suggest, would you expect the crab shells to be slightly less durable in Bellingham Bay than some other areas?
I have no idea what will happen to crab shells. But thank you. I'll ponder it. It'll keep me awake tonight. Thank you all.